Naku, ano ba naman yan? Ulan na naman.
Kaya nga eh. Kamusta na pala yung research mo?
Ayun. Ang hirap talaga. Wala pa rin progress up to now. Oh, eto nga pala yung buka. Salamat, laking tulong talaga niyan para sa research.
O nga pala, nabalita mo ba yung tungkol sa research ni Emma? Ano ulit tawag doon?
Yung about sa... ayun! About sa pagmanipulate ng mga genes ng mga organisms.
Organisms? Kasama ba halaman doon?
Ang alam ko, oo. Ay, edi, pwede rin yun magamit sa mga tao, no? Pwede rin siya maging beneficial sa mga tao. Hala, edi sana magamit din siya sa mga medical field, no? Para magamit as a cure sa mga taong may sakit. Lalo sa mga diabetic, katulad ko. Ano ba yan? Kasi yung bago kong gamot, ay nako, nag-a-allergy ako doon. Ay, buhay. Kaya ko nga ginawa tong research na to para rin naman sa sarili ko. Tapos, up to now, wala pa rin progress. Forever na ba akong deprived sa mga sweets? Grabe, gusto ko talaga makatikim ng cake.
Ako nga rin may problema sa trabaho eh.
Oh, bakit? Anong ganap?
Sa tingin ko dahil to sa mga peste na kumakain ng mga tanim ko. Ang hirap na nga magpatubo, wala tuloy ako maangkat.
Ay, tamo, ang hirap na talaga mabuhay ngayon, no? Parang dati, ang dali-dali lang maghanap buhay. Tamo, dumadoble kayod ka na sa pagtatanim. Dala ng internet na yan. Ano ba kasi yan?
Oo nga eh, dahil to sa internet na yan. Ang dami-daming nilalaman, puro naman di totoo. Speaking of this libro, idada kaya natin to kay Emma. Total, malapit lang naman dito yung office niya.
O sige, tara.
Tara.
Tara, Nosy.
Sana namin tong libro mo.
Sige, palagay na lang dun sa... Wait. Hello? Opo, sige po. Diyan na po, noted. Bye-bye po. Guys, go ahead. May papapasa sa akin si Doc Nuts na Doc Nuts. Guys, kayo na bahala muna dito, Bye-bye!
Dito ko na ba kaya ilagay ito?
Sige, sige.
Uy, ano to?
Uy, huwag kang malikot. Hindi natin to office.
Okay lang yan.
Hi everyone! We are the Three Little Scientists!
Where we hold the knowledge of the
Past, present,
and future.
with the help of science and technology. We have the...
Biotechnology.
And we are going to talk about...
Genetic engineering. Since long ago, humans have already begun engineering genes for centuries. From plants na kinakain natin, pati na rin ang mga hayop na ating nakikita. At dahil sa CAS9 na nagsisilbing gene scissors, they can now target, remove, and replace any stretch of the double-stranded DNA molecule with any living things. At nang dahil sa discovery ng CRISPR, mas napadali, napabilis, subalit magastos pa rin ang genetic engineering.
Lahat ng organisms ay may iba't ibang physical qualities at characteristics. Would it be possible to extract these qualities and exchange them with the desirable qualities of other organisms?
Abay, pwedeng-pwede. Through genetic engineering. Well, ano nga ba yun?
Genetic engineering. Tinatawag ring genetic modification is the alteration or manipulation of an organism's DNA in order to create a newer gene framework with desirable properties. It is either nadadagdagan or nababawasan ng genes from it. Tinatawag rin na transgenic or genetically modified organisms, mga organisms na mayroong one or more of their genes altered.
Actually, maraming mga hayop ang namamodify for some specific reasons. And ginagamit rin ang genetic engineering for agriculture sector. So, where does genetic engineering find its use in today's world?
Hindi mo lang namamalayan, pero maraming bagay na ginagamit natin sa araw-araw ay resulta ng genetic engineering technology, tulad ng yeast.
Through the advancement of biotechnology, nakakatulong ang genetic engineering by modifying microbes and plant-based materials.
Additionally, energy crops can now be bioengineered to grow quickly na nakakatulong to produce huge quantities of biomass na pwedeng i-convert into fuels, oils, alcohols, diesel, or other petroleum products. Then, energy can be created from this waste.
Lastly, majority ng GMO animals are made para sa mga scientific experiments. Mga animals na ito ay ginagamit as models para sa mga researches regarding how specific genes function, frequently in relation to how the genes impact health and illness.
Ngayon, aalamin natin ang process of genetic engineering, taking insulin as an example.
To produce significant amounts of human insulin, the insulin gene is introduced into a bacterium. With the aid of specialized enzymes, known as restriction enzymes, the human insulin gene is located on chromosome 11 at position 15.5. The insulin gene is removed from chromosome 11 using special enzymes called restriction enzymes. Bacterial cytoplasm contains tiny circular strands of DNA. These strands of DNA are known as plasmids. A plasmid is taken out of the bacterial cell. The same restriction enzymes that were used to remove the insulin gene from the human chromosome are utilized to cut open the plasmid. In places where one of the two DNA strands is slightly longer than the other, the restriction enzymes leave sticky ends.
Since both the human DNA and the bacterial plasmid were cut using the same restriction enzyme, the sticky ends are complementary and can be joined using complementary base pairing. Sticky ends are utilized to attach the insulin gene to the plasmid, and an enzyme called ligase is then employed to seal the two pieces of DNA together. After that, the altered plasmid is once again injected into the bacterial cell. Because the altered bacterium also has some human DNA, it is referred to as a genetically modified or transgenic organisms, GMO. Its bacterial DNA is referred to as recombinant DNA. Then the human DNA will be recombined with bacterial DNA. The bacterial cell is then put in a fermenter which creates the ideal environment for quick asexual reproduction with the right temperature, pH level, and quantity of food.
The offspring are all clones of the original transgenic bacteria because the bacteria reproduce asexually. They all have identical recombinant DNA. In order to survive, the bacteria express their own genes.
In other words, they produce the usual bacterial proteins as well as human insulin by expressing the human insulin gene. The fermenter has developed into a society of little factories that are all making human insulin.
After that, the insulin is removed, refined, and packaged. Humans can now inject themselves with human insulin.
However, there are a few risks associated with genetic engineering. Isa na doon ay ang mga modified food that may contain hidden allergens. Additionally, kapag nag-combine ang DNA ng dalawang magkaibang organisms, it can change a non-allergenic meal to an allergenic meal to some people.
Kaya naman, dapat lang na mag-ingat tayo sa pag-conduct ng mga experiments. Research must be thoroughly and carefully managed to prevent long-term loss of life or resource use. However, in the end, we have to acknowledge that no technology is ever perfectly secure. Fortunately, genetic engineering hasn't yet had a catastrophic result.
Let's hope we can maintain it that way and use this great technology to change the world for the better.
Ang teknolohiya at hagham ay patuloy na nagbabago para sa ikabubuti ng ating kabuhayan.
Kaya't mainam na pag-ingatan ng mga ito at patuloy na pagyabungin.
And that's all for genetic engineering.
Till next time!
Bye!
Bye!
Ano yun?